Good morning, everyone. Today, in view of World Technology Day, we are celebrating Swachhata Pakhwada Day uh, on arranging a lecture on biological waste and its disposal. In the very beginning, I would like to invite our Dean Faculty, Professor Ranjan Burman, Senior Professor, to please inaugurate the day.
Okay, good morning and welcome everyone. We are here to celebrate the World Technology Day or the National Technology Day to be more precise, which is on 11th May. Uh, and this is to celebrate the successful nuclear test in Pokhran in 1998, as you may all remember. And it was established by the then Honorable Prime Minister Sri Atal Bihari Vajpayee ji to sort of uh, emphasize the importance of science and technology for the nation building, and it has been celebrated since then. And today, to celebrate that, we are discussing something on biological waste and its disposal. Many of you know that, I think all of you know that there are different kinds of biological waste which should be handled carefully. These are like sort of plant waste, food waste, animal and human waste, and more importantly, the biomedical waste. And all of these should be handled carefully, and their disposals are very important. And today we have a speaker, Dr. Amarnath Pal, who is going to elaborate on this. So thank you very much.
Thank you very much, sir. Now, I would like to request Dr. Amarnath Pal, who is our today's speaker. He is postdoctoral research associate 1 under Dr. Subhashish Haldar in the Department of Chemical Biological Sciences, and his specialization is in cancer and stem cell biology, and he has done his PhD from the University of Nottingham in the year of 2019. So, Dr. Pal, please come up on the stage.
So, good morning to all of you, and it is a great pleasure standing before you all to deliver a talk and discussion about the biological hazards and its management, and at our mechanotransduction biology lab established by Dr. Subhashish Haldar. So, the point is, yeah, before I go into the details that what are the biological wastes and how we actually manage it, first it is important to know that why cleanliness is in laboratories and is important, because for my means it is my theory that the integrity of our work actually begins with the cleanliness, and the cleanliness in the laboratory gives the consistency of our experiment, gives the reproducibility of our data. Okay. Without cleanliness, doing science is just next to nothing. So we need to emphasize on the cleanliness, and we have to be very much sensible, and we have to be very responsible scientists to dispose the biological hazards what is generated during the time of experiments. Okay. In the beginning, it is very important to know that what kind of studies we are actually conducting in the mechanotransduction biology lab. The mechanotransduction biology the philosophy behind this lab or the experiment or the research work is the downflow of the single molecule studies, because so far whatever the information about the progression of the disease we discovered, it is completely based on the cell and molecular biology lab. But being an advanced breed of primates, we always try to pursue to answer some difficult and critical questions in the experiment through the groundbreaking experiments, that how the disease progress. And because in order to develop new therapeutic regime. So here in single molecule studies, we are actually revealing the dynamic nature of the molecules, what is happening inside the cells. Cells related to any kind of diseases. It could be cancer, it could be diabetes, it could be the autoimmune disorder, or any other advanced diseases where the uh, promising therapeutics is not existing yet. So this technology we are through the acquisition of the new technology, that is the single molecule studies. We are actually trying to reveal that how the protein behaves, because in our evolutionary theory there are number of mutations, there are number of alterations happens in the proteins, and proteins is the functional unit of life, because it drives any cells to survive, it drives any cells to become a pathogen. So this technology actually help us to understand that how the alteration in the functional protein involves in the progression of the disease. And one more important, the most, I would say the foremost important part of this technology, of this lab, is we are isolating, we are studying a single molecule at a time. Because in a cells, there are millions of proteins actually generating, degradation is happening in each an hour, maybe in a day, or throughout the year, or throughout the years. So here we are actually in this technology, we successfully isolated one of single protein, one single molecule, and we analyzes it. We do the studies that how this alteration is actually responsible for the progression of the diseases, and how it contributes to the development of the new drug molecules, which can be clinically applicable. Because we know that cancer is a dangerous disease, it is a very lethal, and people, means the patient who get affected with the cancer, he dies every day by thinking that the patient doesn't have the enough time to survive. The other part is that family of the patient is also gone through a severe stresses. But here in this, we actually projecting, we trying to answer some unanswered questions so far in the cancer, that how the transcription factors, that is the trigger factor actually inside the cells, that drives the cancer from benign stage to the malignancy stage. So in these single molecules, we are projecting our study in such a way to understand that how these molecules are affecting the progression of the disease. As I already explained here, the cancer mechanisms, that how the detect mutations altering protein functions driving tumor growth, because there are numerous. So far we actually detected, the scientists detected, even we also studied in many of the proteins, we know many of the proteins, that how this means mutations in these proteins drives our cancer growth. And so, and how it actually affecting the, or altering the normal signaling pathways associated with the normal cellular behavior. But yeah, so along with these studies, the whole studies of of these molecules in a single molecule platform, what we can achieve, we can achieve the detection of the biomarker, novel biomarker, because these biomarkers is very important to means it helps in the prognosis of the disease. The clinicians, the surgeons, the doctors, the scientists all depend on the expression of the biomarkers in the cancers or any other kind of diseases, of course, because with the help, these biomarkers helps us to understand that how the patients or the tumor or the cancer is responding to the given therapeutics. If we so this single molecule platform may help in the detection or discovery of the biomarker identification. It is actually very novel thing here, because in many cancers still the biomarkers is not known, and that's why it is very bewildering for many doctors and clinicians to come to the conclusion that what is the long-term survivability rate of a patient. But if we actually succeeded in developing or in discovering the new biomarkers for the cancer, then it will be the awesome thing, because it can be a groundbreaking research and it can revolutionize the therapeutic regime in cancer. Target interventions. I believe that most of us actually heard about the targeted based therapeutics. We know that cancer has a standard therapeutic regimen, that is the chemotherapeutics. But in the single molecule platform, we can bring the innovations in discovering the targeted therapeutics. It means cancer cells must have some signature, and we can actually target that signature for a betterment of the patient prognosis. And the bridging the disciplines. So combining this molecular biology with oncology, we can discover a novel therapeutics regime. Yeah. So three of the things in a marker we are actually targeting through this in our mechanotransduction biology lab. But every research comes with cost, definitely. Okay, so here we also, whatever the experiments we do in the cell and molecular biology, in the single molecule platform, they are actually carrying some sorts of biological waste. And the most important part in along with doing the good science, it is the responsibility for us that how we are actually segregating the biological wastes and how we will going to dispose of. Because being a scientist, it is, I believe that it is not only the responsibility for us to do the good science, but at the same time it is our responsibility to take care of our neighbors, of our friends, of our families and the colleagues, so that they don't get affected with any of the diseases or the pathology. So biological hazards or biohazards are the biological substances that pose a lethal threat to the users or the people surrounding us. So these hazards can cause, we actually categorize it, that these hazards can be different, is classified into the, can cause, means infections, allergic reactions, toxicity, or even death. So these are the four categories is really dangerous for the user as well as the neighbor. So we have to be very much sensible while doing the research work. So we categorize the biological hazards in four different categories. One is the bacteria, we all know. Viruses like HIV, hepatitis B, influenza, that causes transmission of and cause the chronic illness. And cell cultures and DNA, as you can know, that is the human cell lines. We are and we grow the cells here in the cell culture, where we extracting the nucleic acid and we for the downstream study of the protein and the prions. We are all aware of these four versions more or less, but prions is very much dangerous here because it is a misfolded proteins and it can cause the neurodegenerative diseases. So because of these four categories, it is very important to maintain the proper SOP, that is the standard operating procedures, to rule out the cause of or the occurrence of the biohazards. Okay, so coming to the point that how can this these pathogens or the material or the chemicals can actually affect a person. So there are four important routes where a person can affect with the biological hazards. One is the inhalation, second is the ingestion, contact and ingestion. So in inhalation in laboratory, in any kind of regular routine laboratory, we use different kind of chemicals, right? And some of the chemicals actually cause aerosols, produces aerosols. So if a user is actually inhaled, then it can cause a serious problem. For an example, while handling the some fumigated means some acids like ammonium chloride or HCl, these are actually very much dangerous for the lungs, because inhalation of the HCl or the ammonium hydroxide or any kind of chemicals which has propensity to cause the aerosols may cause the COPD, that is the chronic obstructive pulmonary diseases. It means it will affect your breathing capability. The second one is ingestion, accidental swallowing of contaminated materials. It is true in the lab, no one actually wants to have it, have any bacteria or any chemicals, right? But sometimes we are human beings, we do some things very irresponsibly. Okay, so we think that we are sitting in the lab, there is no work is happening, let's have an apple or banana, you are eating it, right? And always keep in mind that this can cause a serious problem. You may not know that what is going inside. So it is very much important not to have any kind of food, especially in the biology lab. Never ever had any food. Okay, even if you are actually consoling yourself that I washed hand with the hand wash, let have a banana, let have the apple, let have the tiffin together, but never do it, because with this ingestion, because with this route, you never know that what the hell is going inside you. So please stop having the food inside the lab. Even don't even try to open your tiffin box. Contact, definitely skin or mucous membrane, because skin and the mucous membrane means the serous layer inside our buccal cavity, inside the esophagus, in the stomach, in the intestine, in the rectum. Okay, so here in this case, sometimes there is a spillage happens while handling, because we are scientists, whether professors, whether students, or whether postdocs. Okay, we are human beings, and accident happen can anytime while handling the things. It is not about that how much experience I have, but there is always a chance to the spillage of the chemicals, because accident happens and it never comes with prior information. So this is one of the things. Suppose I actually, honestly speaking, I can give you the one example what I observed, because I did my PhD in the year of 2019, and after that I worked in an industry. I saw a candidate in the pharmaceutical industry. Actually, I saw a chemical who who actually very much confident about handling the chemicals, because he is experienced. I am not taking the name of the company, but it was a big issue there. And he somehow, I will not take the name of the chemical as well. It is not ethically good, but I'm just giving an example. There is a very dangerous chemical was kept, and he was unaware of it. But he was very confident that how he handles the things. And accidentally, it actually spilled on it lab coat. He was wearing the lab coat. I saw it in my eyes. Believe me, it spilled down directly on the stomach part. Okay, and it gave him an immediate third-degree burn, even wearing the lab coat also. So while handling the things on the bench, no matter how experienced you are, be very careful. Don't use any chemical with a great confidence. As I said, accident can happen. uh, so be very careful, wear the proper PPE, and try to avoid the arrangement of the glass bottles in front of your hand, because sometimes, because we all means do the experiments in a certain deadline. We know that this is the time I have to finish the experiment, this is the time I have to write the paper, this is the time I have to go through some other places for the experiment, because we all jump around, right, in the doing experiments. So while working on the bench, keep your bench neat and clean. Don't put the glass bottles in front of the hand. Okay, place it in a proper place so that while working there is no such accident happens. The fourth point is the injection. Yes, I know that in the particularly in our biological science, we do loads of uses of the injection. Maybe other stream also using those injections to plunging the chemicals in a certain instrument or something. Okay, so but these injections is very dangerous because while working with the injections, it can prick your finger. Maybe in the injections there is a drug molecule is there inside, or maybe there are other chemicals maybe inside. So if you while handling it, if it is actually prick into your finger and if you push it, it can go to the intramuscular or the intravenous, and it can cause a serious problem. So please handle all these injections material or the serpents in a careful way so that it won't harm you. Okay, so doing science is good, but at the same time, doing science in a good environment is also much more important. So here, scientists actually, in whether it is in the clinical practice or whether it is in the academic or whether it is in a BSL4, means high class research laboratory, okay, these symbols actually are very much important. These symbols are used throughout the world. So there you can see that the biological hazard, it's a symbol that actually a family. Let's say that is a biological hazard. This symbols is actually representing the biological hazards. But under that, you can see this little sign. Okay, this little sign is here for the BSL1, BSL2, BSL3, and BSL4, but there are different colors code. Okay, depending on this colors code, it is actually showing that what kind of biosafety we do require while working in the laboratory. So this biosafety level 1, I highlighted them in the yellow color. You can see that in the biosafety level 1 laboratory, it indicates that it doesn't cause any disease. Okay, in healthy adults, of course. Please quote it healthy adults, because in healthy adults, we have a much more stronger immune system compared to the infant or the little one. Okay, so but have a minimal potential hazard to the environment and the laboratory personnel. Okay, and there is an effective treatment is available. So when we work with these chemicals which satisfy this criteria, actually fall under the biosafety level 1. For an example, let's say Escherichia coli or E. coli, the bacteria, it comes under the biosafety level 1. What is biosafety level 2? It is actually the presence of organisms capable of causing mild infections, having moderate potential hazard to the environmental laboratory personnel. It is fine. It's okay. Okay. It can cause the moderate potential. It carries some moderate, potential hazard. It has to the environmental laboratory personnel, but they are not able to form the aerosols. It doesn't come into the air, so that there is a very less chance it goes inside your nasopharyngeal portion to infect you. Okay, and having effective treatment or preventive measures. Doctors know how to handle it, because if I get affected with such kind of things, then and if I report to the medical team, they know how to deal with it. They have the medications, they have the injections, they have the proper treatment regimen. So if anyone get affected, nothing to worry about. Within 72 hours, the patient the affected person will be stand on his or her feet, and they will be happy. Okay. So biosafety level 3 is the presence of organisms that capable of causing severe infections, okay, and spreading via aerosols. For an example, let's say Mycobacterium tuberculosis is one of the bacteria that cause TB, that affects the respiratory system. Okay, these organisms are very much dangerous. They are actually these are harmful for the person who is actually handling it, and but it has a moderate community risk, because I'm working, let's say, and if I inhaled it, then it will affected me, but not the other people around. So it is not a dangerous part for the community. Okay, and it has a prevention measure is available. We know, for example, micro TB. There are medications are available which actually helps in the prevention of the occurrence of the TB. It can be curable. Okay, but it's a long-term treatment. Okay, so we need to be very careful while handling this kind of organisms, and this kind of organisms should be handled in the biosafety level 3 environment. And the foremost and the most dangerous, there is no treatment. In the BSL-4, the conclusion is death. Okay, so here there is causing serious infection to the individuals and communities. If I get affected, my community also get affected. There is no such treatment is available. Okay, and most of the time it is a death. So here in the BSL-4, they work in a very containment zone. Okay, with the proper PPE, means personal protective equipments, and maintain it. Their SOPs are really very high. This protocol is very high. Even they means I don't know, means in industry also they don't allow PSL-4 in a very specific industries all over the world. If I'm not so wrong, I don't know whether it is increases yet or not, but all over the world only there is a 40 or 50 BSL-4 lab is available. Okay, and suppose for an example, what kind of organism? This is a dangerous viruses. For example, the coronavirus is one of them, which actually get leaked from the BSL-4. And and we all are aware that what it happened. There was no treatment. There is a endless death happened. Okay, so this one is just hell. Okay. So we can understand that depending on the possibilities of the diseases, depending on the what kind of organisms we are handling or the chemicals we are handling, this measurement we actually took the level of biosafety, that is biosafety from 1 to 4. So before coming to the BSL-1 and BSL-2, okay, here you can understand that we don't work here, our microtransaction biology lab, we don't here work with the BSL-3 or the BSL-4. Our every work covered or comes under BSL-1 and BSL-2. Means from the definition itself, it is showing it is completely safe. There will be at least I will say 99.9% chances there will be no hazards will be there. Okay, we are all in safe, whether it is personal or whether it's communities or whether my friends or families, everyone is safe. First thing first. Okay. So to make this place more safe environment for the work, we do means we further give the definition that what is the difference, what is the major difference between the BSL-1 and BSL-2. So as I said, it is actually suitable for work with minimal risk agents like non-pathogenic strains, as I already said, and here it handles moderate risk agent like attenuated viruses and mammalian cells. Okay, attenuated virus, you may actually get concerned about the viruses, but these viruses have no potential to cause the diseases in human, and actually these viruses are completely under control of the CDSCO, that is Central Drug Office, and it is absolutely fine. So here we use the standard microbiological practices applied, as there we established it. No special containment equipment required. We don't need any special kind of hi-fi, instruments or something like that to protect ourselves from the infections. And what can be performed on open bench? In open bench also it can work. There will be no chance of infections at all. But still, we use the cabinets. Typically, they say that no biosafety cabinets is mandatory, but still we work all these things in a biosafety cabinet, in a lab, simple lab, is a laminar air flow chamber. So it is not only minimizing the risk of the biohazards, but it is also eradicate the possibilities of the biohazards. So it is completely safe. There is no chance of infections. Here, BSL2 characteristic, as you can say, that we are using with the viruses and mammalian cells. Okay. So here it is the access restricted and controlled. We have the clean room there, but we are now standardizing, but not started our work yet, to understand that there should not be no infections, there should not be no contamination, and it is the access restricted. Okay, why? Because we should not just go around with the human cell culture, because cells are the unit of life which is inside our body. But here, generally, scientists actually culture, they grow the cells outside the body. That is called the in vitro cell culture. Okay, so and they are very much prone to infections. We will not get infected, but cells will get infected from our end. So that's why it should be it should not be just accessible to everyone. Only the users are only handling or operating in this containment zone. Okay, and we use the obviously we use, because we go outside, we come from home to the laboratory, so there are loads of dust particles actually in our body, in our costumes. So if we actually go directly with wearing these shirts inside, then without our notice, there are thousands of molecules, millions of molecules is actually going inside that zone, that actually affecting the experiment quality. As I said, that reproduce with the experiment good and consistency result only comes from a cleanliness. So we need to keep the zone very, very clean. Okay, so we need to use the PPE, of course, and here is the enhanced laboratory practices to avoid exposure. So we need to, because since we actually working on this cell culture for a long time, we know how to handle it. There is a particular SOPs are there. There is standard operating procedures are there. So we are actually developing the procedures now. We are actually doing the SOPs. We are actually drafting the SOPs, that how to behave inside this BSL2 lab. And obviously the cabinets here, there's a hood that is called the class 2 biosafety cabinet is required, because just like this, with no biosafety, means in the open bench here in the basal one thing, we can do the experiment in the open bench, but here we can't, because it should be inside the biosafety cabinet, which is the most sterile environment, which provides a sterile environment to the system. So the question is that what is the class 2 biosafety cabinet? Class 2 biosafety cabinet is actually made in order to protect the user and the environment. Okay. So here it has the HEPA filter. That is the magical thing. HEPA filter means it is a filtration unit. It calls a high efficiency particulate air with the pore size of 0.22 microns. So 0.22 microns, as you can understand, there will be no bacteria, nothing. It is completely free. Okay, and work is performed in a ventilated and enclosed workspace to contain biological agents. You can see this is a biosafety cabinet, and here is the working space. Here we work inside here. Cells should be cultured, nurtured, whatever experiments we will do, it will be inside this hood. Okay, and here in this part, there is a HEPA filter. Two level of HEPA filter is here. I will explain that how it works, so you can understand the mechanism here. Okay, so the conclusion is no contamination again. And this, the most important, I almost forgot to tell that once the air is actually filtered here, in a whole room, it is only get the purified air itself, the much more pure, high class purified air. So there is no chance of inhalation of the aerosols or the infectious agent. So this is the mechanism how it actually, the class 2 cabinets actually purify the air. So as you can see, it's a side view of the hood, and this is the working bench inside the hood. Okay? And here is the portion where we only just put insert our hands for the operating the experiments. Okay, so the room air enters, and you can see the pump. It's a fan, so it creates the vacuum inside it. Okay, so once the environment air actually enters, it directly goes down. It doesn't hit in the workbench. There is a net, it directly comes here and goes up, and this fan creates a high pressure of the air, and these two are the HEPA filter with a 0.22 microns pore size. So once the positive pressure actually creates by this fan, the pressure, the HEPA filtered air. What I said, that this thing in the room also, it actually circulates the pure air, purified air. It filtered the air, and some portion of the air goes inside. Okay, so now you can see, since this portion of the air is filtered through this HEPA, it means completely sterile. So it's a very simple mechanism, but very effective. So this is why I am saying the person who is working here is completely safe. Even the samples for the cell culture, even it is also safe. There is a very less chances of the contamination, unless and until we just go inside outrageously without wearing the proper PPE or malpractices inside the lab. Okay, or else this place, this cabinet actually creates a very safe environment for the user and the environment. Okay, well, so where we actually keep this is only biosafety cabinet is the last resort to keep the environment clean, or to make the happy place to work. No, we actually create here the means ISO 7 and ISO 8 standard clean rooms. Okay, so in the ISO 7 clean room, you can see that it is so much fascinating and so precise that in ISO 7 the room only contains 10,000 particles. Okay, that is greater than or equal to 0.5 microns per cubic foot. So 10,000 particles is nothing. Okay, so this kind of particles, it continuously actually purify the air. So it gives another level of purification of the purified air inside the ISO 7. So it is again gives a very controlled environment where means we're just eradicating the chances of infections in this ISO 7. Okay, and obviously air is filtered through HEPA systems. So HEPA filter is also present in the hood and also present in the roof of the room, so that every air is completely out of the viruses, out of the pathogens. It is completely safe for to safe place to work. And obviously, if the air is actually free from bacteria or any pathogens, obviously it supports the stringent contamination control. Compared to the ISO 7, we have the ISO 8. Okay, so in ISO 8 clean room, it has the little bit of higher particles, 1 lakh particles. Still, it is nothing, because here in ISO 8 we don't do the cell culture actually. Here we used to work with the microscopy, centrifugation, vortexing. Okay, all these things, because for those microscope we don't need that kind of high level of contamination free zone. It is just a microscope, it is just a centrifuge machine. So 1 lakh particle in the ISO 8 is quite tolerable. But the thing is, for both of them actually doesn't allow for the aerosol mediated pathogenesis in that case. Okay, and obviously it provides a basic clean environment and easier and cheaper to maintain than ISO 7. So, and of course we in our SN BOS we actually establish this, both of the clean rooms there. Okay? And foremost thing, only two persons are allowed here in this zone, because if we actually gather around too many pupils, then again the particle number will increase and the chances of the contamination will increases. So 2 person at a time we allow generally. So safe handling practices, we all aware about this thing more or less from the time of corona. Okay, we wear the personal protective equipments, we wear gloves is mandatory, eye protection, goggles or face shields, because while working in the hood or ISO 7 we should not talk, we should be very much well controlled person, we should not talk, because once we actually talk, we spit the we spit unknowingly, it can goes inside the hood, it can comes in the contact with your cells. It is means in this ISO 7 or ISO 8, if there is any contamination happens, it is completely fault of the users who is handling it, it is nothing to do with the environment. Okay, so it is very much important. Goggles, face shields, and respirators or fume hood for aerosol risk. Of course, for our kind of work, we don't use any kind of aerosolic generating agents. Okay. And technique and equipments, careful pipetting to avoid aerosols. Class 2, again, I'm emphasizing this point because we need to know how to handle this class 2 biosafety cabinet. And we have the proper dispose of the surf disposals. We use the needles, scalpels, blades. We need to keep it in a proper containers, because only the surface things we need to actually incinerate. Okay, so we need to incinerate in order to completely uh, destroy the particles there. Well, so and the disinfection and sterilization process is very it's very important here while working in the biological system. And we have the autoclaving. You can see there are 121 degrees centigrade for 15 yeah, exit. Okay. 15 psi for 20 minutes for the sterilization we give, and the chemical disinfectants we do the bleaching or the 10% bleach, ethanol, for the rapid and effective cleaning of the surface. And the equipment just and the regular bench top we use to clean the hood bench top regular basis with those chemicals to get rid of any kind of infectious agent. This is the waste segregation disposal workflow. While working, we have to very much aware that we need to separate the biohazards. While doing the work immediately, we need to know what are the solvents, what are the chemical waste are there, and we have to dispose of immediately in a particular or allotted dustbins. Use proper containers, as I said, with the proper labeling and dispose safety, autoclave, incinerate biohazards, and it is most important, we need to label the containers so that people know that what kind of hazards is there inside the container, because every container doesn't contain the same kind of biohazards. So I just give an example. These are some of the symbols or the stickers we can actually paste on the waste bins, depending on the what kind of biological hazards we are actually disposing there. Okay. And this is the emergency procedures. As I said, there could be the spill response, there could be the exposure incidents, or as I already explained this, that what are these things. And but this thing is actually very important, and this part is actually very important to know for each and every users in the laboratory. That is the MSDS, that is the material safety data set. Because in this material safety data sheet it is available in the website for each and every chemicals, so we need to download it, we need to keep a proper file for it. Suppose for an example, in many times we work in the lab alone, okay, and some spillage happens, something happens with the chemical accidentally. So if you have the MSDS ready, so you know how to protect yourself. So you won't run around here and there for the predictions. If you have the knowledge of the chemicals, you also know, because in MSDS there is a very cleanly written. If there is something accident happens, what you need to do and what you don't need to do. So this file, maintaining this file is very important in the laboratory. Regulations and guidelines. So these two are for the industrial level. I just give a simple example. There is a bloodborne pathogens. If you are working, this in academic it doesn't require. Okay, and the CDC or the NICMBL is for the biosafety guidelines for lab practices. Under this license, this both license comes. Okay, but for the bloodborne pathogens, it is required for the industrial platform. But IBC is equally entertained in the academic as well as for the industry level. It is a institutional review and approval of biological use. We should know, everyone should know that what are the biological hazards are there and how to dispose it and what kind of hazards it can bring while doing the experiment. And so in order to mitigate the risk, what are the best practices? There are four golden points. One is the training. We need the proper training. We need to give the proper training to the newcomers in the lab. We need to draft a proper SOPs, that is a standard operating procedures, so that people know how to handle the things inside the lab. Safety culture, that is, we need it is not the only means it is not the sole responsible for the users, but it is a responsibility for the other people also working in the lab. It's a teamwork. It's not a only single people thing. It is a teamwork. So we need to be very much careful. We need to grow up. We need to nurture the culture of the safety protocols. Okay, if someone actually spills chemicals or something happens, we should not make our sentiments in such a way that he or she did it. Why should I clean it? Just clean it. Okay, it's a teamwork. Okay. and resources, yes, access to updated guidelines supports risk management. Definitely, yeah, the officials are there. They must actually have the updated guidelines. They must have some updated guidelines actually provided by the governing body. Who is responsible in a workplace? Each and every people, not only the users. Each and every people. It is the responsibility for the employers, employees, safety officers, institutional biosafety committee. Rules can be made easily, but the point is followers are very less. So we should be very much aware about these things, and we should follow the rules in a unitedly. Okay, to keep the workplace safe, to keep environment safe, and then only we can actually nourish the science for the human welfare much better way. And conclusion, next steps, key takeaways, key takeaways is, as I said, understand the hazards. What are the hazards is there? What are the exposure routes? Apply strict safety and handling practices that comes under the SOPs, that is a standard operating procedures. Follow regulations and emergency protocols. Okay, and what is the next step? Next step is engage in continuous safety training. It is just not just speaking and forget about the things. It should comes in the practice every day in our routine laboratory practice. It should incorporate. Okay, regularly update risk assessments. We need to assess that what kind of risky things we are doing. Today I am using chemicals. Tomorrow I will going to use the cell culture. Day after tomorrow maybe I am working with the bacteria or the viruses. So we need to understand what kind of virus I'm using, what kind of bacteria we are using, what kind of cell culture means cell culture we are performing, what kind of genetic engineering we are performing. So we need to understand that what kind of experiments we are doing and what risk it is actually carrying while working, okay, in the laboratory. And foster a culture of personal responsibility. Once you are working in the lab, so it is your responsibility, whether you are wearing the lab coat or whether you are going to need the goggles. Because every day there are 8 people working under a supervisor. It is very difficult for the supervisor to come down to the lab and see who wears what. It is the company's scientists or the PhD students or the postdocs' responsibility that whether they are maintaining or fostering the proper PPE or they are doing the proper risk assessment. So don't blame supervisors if anything happens. Okay, And thank you. Thank you very much for your consideration. Yeah.
If anyone have any question.
Okay. Yeah.
Nice job.
Oh, thank you.
Thank you both. So just a clarification, Ravi.
Yeah, sure.
So you said that you were doing some cell cultures.
Yes.
And what kind of cell you were dealing with? Like, is it like normal or cancer cell?
Both.
So then, is there any possibility that those from those cancer cells one can get cancer?
Yeah. Okay. So it's a good question actually. The point is that here the cells cannot fly. Okay, first thing, because these cells are all adherent cells. We culture them inside the flask, in a filtered flask. Those, if so, there is a very less chance that the cancer cells crawls and comes out from the container and get inside your body. Means it doesn't happen in that way. If it did happen before, then I would not stand here. I would be in some cancer hospital and waiting for that. So it does never happen in that way.
Sorry. So let's say if there is an accident and a spillage happen and it like fails in our
Yeah.
skin or something like that, then what?
Okay. So the point is that suppose for an example, the cancer cells actually adhere on the surface of the flask. Okay, but we nurture the cancer cells in a particular medium. Inside the cells, inside our body, there is a blood and the lymph. We actually nourishes the cells. Similarly, cancer cells also inside the flask actually nurtured by the media. It is a liquid media which provides the essential amino acids, nutrients, oxygens, the CO2, and maintain the buffering system. Okay. So if there is spillage happens, the liquid will come. Cancer cells cannot come. Okay. And if accidentally happen, it can happen. Okay. If accidentally happen, we use the bleach, 10% bleach. Just take it, pour it as much as possible, wipe it off, drop down. These are only 5 minutes job. So, and after that, you can test it with the microbiology testing or some other clinical testing. You won't find any cancer cell or the remnants left inside. Yeah.
So actually, we are actually expert on cell biology and cancer, all this stuff. So, in answering Subha's question, is actually absolutely no chance of getting cancer because there are many steps we have to follow. So that there is almost no chance that it can go inside our body and it can cause disease.
Zero chance.
Yes.
Zero.
So one of the way that Omar said that okay, so it cannot go out of the media. That's going into media. Of course, when the cells go out of the media, there is no food. So
They will die.
no.
They
So
will
that's
die.
a one. But we are not trusting only this. We are doing a multiple layer of check. So we use one of them is the bleach. So immediately the cells will die. Then we use different type of
Means concentration of the alcohol.
70% alcohol. So there is actually completely clean the whole system. Like when the coronavirus came, what we did, we used the 70% ethanol, and also it in different form like gel type of form. So actually this is a ethanol. So it's clean everything, ethanol or isopropanol. So there is no chance of any biological
Exactly.
material that can go. Even if it pass all this level, if it go inside our body completely fight with them because our defense system already activated. So it will definitely kill them.
And
So there is no chance for.
well, so in continue of this thing, I would suggest that if there is any injection, it is a very rare chance. But if it goes inside, the cancer cells cannot get absorbed through the skin. It is only possible if anyone drink it or if anyone inject it.
Yeah.
Okay, so
Yeah,
actually
if somebody want to try with the injecting it cancer
Yeah.
cell in the body.
then it is a different
So,
thing.
and the immune system don't fight with the cancer.
Yeah.
then these cells will grow and get cancer. But
That is the problem of his immune system.
nobody
Yes.
is getting mad or
Now, one more, there are other part of the
Kind
thing.
of thing
You know,
that,
these cancer cells, these are cancer cells, is a very less chance to colonize inside the body. Okay, yeah, it's very less chance. But if anything happens, there are remedies there. Because I didn't go that much of inside of it, but there are remedies. So what place is completely safe.
also for the if we work with the bacteria and viruses, those are what we are working is nothing. Those are completely safe, completely non-infectious.
Non-infectious.
So if we work on any kind of infectious material, we need a lot of permission, and also we have to build another kind of safety level. Then we can think about that. So for now, we don't have that much license and the safety, so we are not going to work with the
We are
infectious.
We are working on getting the licenses actually. We are working on it. So anyway, so apart from yes, please.
Say like somebody who is experimenting on this cancer cell and have some wounds on his hands
Yes.
or whatever.
Yes.
Maybe sometimes we do a little prick for sugar test or
Yeah.
this. If this cancer cell somehow spilled on this wounds or like
Yes.
that.
Okay. It's a very good question actually, madam. The point is, look, we wear the gloves. Okay. These gloves are nitrile gloves. Okay, and it is made for that. So if you, it can happen. Suppose we are cooking in the home and it gets an open wound, right? So at that time, my request, don't handle it. Yes, because that is the best way. If it has the open wound, no matter wearing the gloves or what, don't handle the cell culture. That is the thing.
Yeah,
Stop.
actually, I was to ask you that is there any daily health checkup or whatever, like protocol in your lab that
No, it
people
doesn't.
who enters in the lab, he has a routine checkup that he has no
No, it doesn't. Look, okay. Here in this trail of the questions, I would say that if, suppose for an example, if anyone has the cough and cold, hot cough and cold, he knows that the user is not well. Okay. Cough and cold affected person should not enter. Okay. Because it may affect or it control, because one sneezing, because if sneezes comes, you can't control it. It's an involuntary response. So if sneezing happens, then the whole room is getting on. So no matter what, ask your colleague to handle the cell culture of that time being. It better not to entertain the people inside. Because if I am sick, if I have a high fever, if I am caught cold, try to not to stay away from that cell culture. Once you are fit, once you feeling you have a proper breathing, means proper breathing, inhalation, exhalation, you are fit, you have no body ache, means clinically you are actually fit. So then only entertain the cell culture, or else no matter how experienced you are, just get off from the cell culture. That is the thing. We should not entertain.
Thank you.
Yeah. Any questions further? Yeah.
Nice talk.
Thank you.
I have just a quick question.
Yeah, sure.
In my opinion, there must be some wastages in biolabs to dump, right?
Yeah.
Is there any standard procedure to do so?
Yeah. Yeah. I mean,
No, I mean in general, which are the standard procedures to dump the wastages from the biolaboratory?
Well, so yes, as I said here, you means I already explained it, that we have a different sorts of waste bins out there with the product proper labeling, that what kind of biowaste is there. Okay. Suppose for an example, in a cell culture, I am talking about the flasks, right? Where we have to do the cell culture. So cell culture flask is a biohazard. Okay. Though there is no cells, but means suppose for an example, what we do to understand this answer, you need to first understand that how we actually handle the thing. Cell has a tendency to proliferate. Means from one cells it becomes two cells become four becomes six. Okay. So it grows exponentially. So we can't only keep the cells in a same flask for a long time. We need to split it. From one flux, we get the 2 flux, from 2, 4, like this way. Okay, we can actually increase it. Now, the previous flux, what we actually, from where we took out the cells to the next flux, that disposed flux should be treated with the bleaching at first, 10% bleaching, and then it should be go inside directly to the autoclave. Once it is autoclave is done, that thing you can check it with the bacterial culture lab. You won't find a single colony in your plate. So in this way, and after that, once you take the things out from the autoclave, just put it in a normal bin, and from there you can dispose of. There are a company who actually handles this waste materials there. Yeah.
Thank you.
Yes, thank you very much. Yeah.
Anyone else? Yeah, I think it is.
Okay. Thank you very much to all of you.
Thank you very much, Dr. Pal.
Thank you.
It is really an informative and very
My pleasure.
interesting talk.
My pleasure.
Thank you for coming.
Okay.